Welcome back to the Learning Studio. This is the Biological Sciences series, and today we begin an exciting new section, Biotechnology and Applied Biology. Our focus is on a groundbreaking tool that transformed science, medicine, and agriculture, recombinant DNA technology and cloning. If you missed our last episode on emerging infectious diseases, the full playlist is linked below in the description. Here's a question. What if we could take a gene from one species and make it work inside another? That's not science fiction. It's exactly what recombinant DNA technology allows us to do. What is recombinant DNA technology? Recombinant DNA, rDNA technology, is the process of cutting and joining DNA molecules from different sources to create new genetic combinations. Think of DNA as biological software. With recombinant DNA, scientists can copy, edit, and paste this software into different organisms, turning bacteria into insulin factories or crops into pest-resistant plants. The basic step: isolation of DNA, extracting the target gene from an organism; cutting DNA, using restriction enzymes, also called molecular scissors, to cut DNA at specific sites; vectors, inserting the gene into a carrier, such as a plasmid or bacteriophage; cloning, introducing the vector into host cells, often bacteria, that replicate and produce many copies; expression, in some cases the gene is expressed to produce proteins, such as insulin or growth hormone. What is cloning in this context? In biotechnology, cloning means making identical copies of DNA, cells, or organisms. Molecular cloning, copying a specific gene. Cell cloning, making identical cell populations. Organismal cloning, creating a whole new organism genetically identical to the donor, like Dolly the sheep in 1996. Case studies and applications. Medicine, recombinant bacteria produce insulin, growth hormones, and vaccines. Agriculture, genetically engineered crops resist pests, tolerate drought, or provide better nutrition. Industry, enzymes for detergents and food processing are produced using recombinant microbes. Research, recombinant DNA is the foundation of CRISPR genome editing and synthetic biology. Case study, insulin. Before recombinant DNA, diabetics relied on insulin extracted from pigs and cows, which was expensive and sometimes caused allergic reactions. Today, recombinant E. coli bacteria mass-produce human insulin, saving millions of lives. Like any powerful tool, recombinant DNA technology raises questions. Should we genetically modify crops and release them into ecosystems? Where do we draw the line with human cloning or gene editing? How do we balance scientific progress with bioethics and safety? These debates make biotechnology both exciting and deeply relevant. Past, present, and future relevance. Past, the first recombinant DNA experiments in the 1970s sparked both excitement and controversy. Present, recombinant vaccines, gene therapies, and GM crops are shaping global health and agriculture. Future, with CRISPR and synthetic biology, recombinant DNA could help cure genetic diseases, fight climate change, and even design new life forms. Why this matters for you. Understanding recombinant DNA is like learning the alphabet of modern biotechnology. Without it, advances in PCR, sequencing, CRISPR, and personalized medicine would not exist. So that's our journey through recombinant DNA technology and cloning, the tools that gave humans the power to rewrite the code of life. In our next episode, we'll continue this biotech adventure with PCR, sequencing, and molecular diagnostics, technologies that detect, analyze, and diagnose diseases at the molecular level. Welcome again to the Learning Studio. Don't forget to check the playlist link below and subscribe so you won't miss the next video. Because once you understand recombinant DNA, you hold the key to every modern breakthrough in biology.